Hello, everyone. My name is Rhett Rotza, and I'm a Field Applications Bioinformatics Support Scientist at PacBio, or FABS for short. Today, I'm going to be talking to you about HiFi informatics for uncovering germline variation. To go over the agenda for this presentation, we're first going to talk about how HiFi sequencing provides you with a more complete view of your genome. Next, we will talk about our available tools for variant calling in both our GUI interface, SMRT Link, and on GitHub. We'll talk about our human WGS variant pipeline for streamlining the use of these tools. And finally, we'll talk about our PacBio compatible partners and additional resources to optimize your analysis workflow. I want to start out by showing you how HiFi reads provide you with a more complete view of the genome. So here we have an IgD pileup view of reads mapped to the STRC gene on chromosome 15. This is a 19 kilobase region. In the top row, we have short reads, which are highly accurate, but you can see uneven sequencing coverage and even complete gaps where we have no sequencing coverage at all. In the middle, we have highly accurate longer reads that provide you with even sequencing coverage, no gaps, and even in those gapped regions that we see above, there's a clear heterozygous variant that allows you to phase these reads into their appropriate haplotypes. And finally, at the bottom, we again see long reads with even sequencing coverage, but with many more sequencing errors, making it difficult to determine what are true variants versus errors. These three rows are SBS, PacBio HiFi, and nanopore technologies, respectively. So you can see how HiFi data provides you with long, highly accurate reads with even genome coverage, no GC bias leading to a more complete view of the genome. And this completeness also includes methylation information, which you automatically get with HiFi WGS runs for free, no additional library prep needed. Like I mentioned earlier, HiFi reads have extraordinary accuracy, and the long read length help you get allele resolution phasing. And this gives you a full picture of genomics and exploring all the variants that occur in your sample. Now, in order to take full advantage of HiFi data and explore your genome and all the different variant classes, you need analytical tools, and PacBio can also offer you that. So while most people are familiar with SNVs and indels, which you can get with short read sequencing, the HiFi genome is a truer representation of WGS, as the long accurate reads from HiFi excel at not only small variants, but also more complex variants, including structural variants, tandem repeats, segmental duplications, and pseudogenes, and coverage across hundreds of millions of bases and over 400 medically relevant genes found in dark regions of the genome, or regions inaccessible to short reads. HiFi reads also enable full phasing of the entire diploid genome and allow you to examine extremely large copy number variants. Finally, with HiFi sequencing, you can detect methylation with no additional library prep or sequencing required. And altogether, this really shows that not all genome sequence data is created equal. We've developed a variety of tools to make the most of HiFi sequencing data, and I'm going to walk you through those tools today, but I'll note that this is just the tip of the iceberg focused on assessing germline variation. And we have many more tools available for other applications like our RNA isoform identification and pharmacogenomics. I'll also note that all of our tools are freely available on our GitHub page. Many are easily installed with Bioconda, and we also offer a freely available graphical user interface called SMRT Link for more point-and-click friendly solutions. Now let's jump in and start to go through some of these tools for human germline variation. First is PBMM2, our read alignment software. PBMM2 functions as a wrapper for Minimap2, which is a standard read aligner for long read sequencing data. So why do we have this wrapper? The big advantage is that it will take native PacBio data from our sequencing instruments as input. By default, our instruments output data in an unaligned BAM format, or UBAM format. And we utilize BAMs rather than FASTQs because it is an industry standard that allows for high compression of the data to keep file sizes small, but with the additional flexibility to allow us to store data beyond just base pairs and base qualities, such as the location and probability of methylation on reads. And while BAM format is an industry standard and can be easily manipulated with software such as SAM tools, Many read alignment softwares like Minimap2 will only accept FASTQs as format as input. Therefore, pb-MM2 is designed to take native PacBio UBAM as input for alignment rather than needing to convert your data to FASTQ first. We've also optimized the parameters in pb-MM2 for HiFi data. Next is Deep Variant, which is our tool for calling small variants like SNVs and indels. This tool was developed by Google Health, which is one of our PacBio compatible partners. This is a deep learning-based variant caller that takes an aligned BAM from pb-MM2 and a reference FASTA file to generate read pileup images. These images enter a classification model, which is a convolutional neural network that scores each potential variant site as homozygous reference, heterozygous, or homozygous alternate, and converts the classification into standard VCF format. For structural variants, we've developed pb-SV to identify break-ins for large insertions, deletions, inversions, duplications, and even translocations within and between chromosomes. The example here on the right is an inversion where the red and blue colors in the pileup image represent the forward and reverse read alignments. Here you can see the HiFi data cleanly mapped to this region, but there's a section in the center where the color changes, indicating a complete inversion that can be detected by pb-SV. Already, we have went over alignment, small variant, and structural variant calling, and these are the most common types of variants examined in WGS data. And we have workflows in SMRT Link for these variant types. For the remainder of the time, I'm going to dive into some of our more advanced and recently developed tools for more complex variants. Let's start with Target, which is our Tandem repeat genotyping tool. Tandem repeats are genetic sequences that are repeated in series. Importantly, tandem repeats have been linked to many neurological disorders such as ALS and Huntington's disease, in addition to being the #1 cause of inherited mental intellectual disability, Fragile X syndrome. Short-read whole genome sequencing offers a limited repeat expansion resolution due to read links being unable to map across these expanded regions. Therefore, to fully utilize long reads to interrogate tandem repeats, we developed Target to provide a best-in-class repeat expansion solution. At a high level, the caller takes aligned HiFi reads along with a bed file, which contains the location information of the repeat expansion in question, as well as the expected repeats from the reference genome. The tool will then output a VCF containing information including the consensus sequence and accurate repeat count at the location, methylation level at each site, motif construction, and the interruption sequences. All of these are reported at the allele level for each haplotype, which is critical for downstream exploration of the mechanism and impacts of tandem repeat mutations on disease phenotypes. To help users better understand the results, we have developed a complementary visualization tool, TRVZ, which will display the events at each location per haplotype, including repeat structure, interruption sequences, and the supported HiFi reads. Here on the right is an example of a sample with FMR1 repeat expansion, where the visualization shows the normal unexpanded allele lacking methylation, as well as the mosaic expanded allele with high methylation, indicative of Fragile X syndrome. Moving on to paraphase, this is our caller specialized to segmental duplication regions. So segmental duplications are duplicated blocks of genomic DNA up to 200 kilobases in length. They are known to be informatically challenging due to high sequence homology with paralogs and pseudogenes, limiting alignment and variant calling. Segmentally duplicated regions comprise approximately 7% of the human genome, and many clinically relevant genes fall into these regions. We often refer to these regions as dark regions, since short reads cannot accurately map or sequence to these regions. However, these regions are often hotspots for structural variations, including deletions, duplications, and gene conversions. So to tackle these regions and genes, we developed paraphase, a tool to fully phase and solve these genes in segmentally duplicated regions using the power of long and highly accurate HiFi reads. Paraphase works by extracting reads aligned to the relevant regions and realigning those reads to the regions of interest with the goal of phasing the reads into their respective haplotypes. By doing so, it solves the problem of alignment difficulty due to sequence homology and allows us to examine all copies of the genes in the gene family, as well as call copy number variants and other types of variants. The output of paraphase is a JSON file containing a summary of the haplotypes and variant calls for each gene family in each sample, and users can use this information to infer disease status and carrier status. To further illustrate the performance of paraphase, here's an example of SMN1 and SMN2, which are genes associated with spinal muscular atrophy, or SMA, which is an autosomal recessive disorder and a leading cause of infant death. This disease is caused by a lack of a functional SMN1 gene, but the copy number of the paralog SMN2 determines the disease severity. Unfortunately, genotyping and determining copy number and variant status for SMN1 and SMN2 is extremely challenging because the genes are over 30 kilobases long and 99.9% similar to one another. So short read sequencing often results in more than 80% of the reads having a map quality of 0 due to the mapping uncertainty. With HiFi data and paraphase, we're now able to determine the number of copies of each gene and the phased variants for each haplotype. Shown in the IGV plot below is the fully phased haplotypes for SMN1 and SMN2. Specifically, in SMN2, we see a deletion of exons 7 and 8 in one of the haplotypes. For larger deletions and duplications, we have also developed HiFi CNV. For structural variants, we've already went over pb-CpG as our break-end caller. However, some copy number events, especially those larger than 100 kilobases, may not have breakpoint signature, and we found that they are often better identified using read depth. One example of an extremely large copy number event without a clean breakpoint signature is trisomy 21. HiFi CNV works by taking your aligned BAM file to estimate haploid depth per chromosome set, and then detects copy number signals by looking for significant deviations from the expected read depth. The output of HiFi CNV is a BCF file containing the copy number variants. However, additional bigwig and bedgraph files are also generated for visualization purposes as shown here. To demonstrate the accuracy of HiFi CNV, we evaluated 25 clinically relevant CNVs from Gross et al. 2019. Of the 25 CNVs, 18 were greater than 100 kilobases and 7 ranged from 13 to 56 kilobases. HiFi CNV was able to detect 80% of the CNVs and 100% of the CNVs that were greater than 100 kilobases. When combined with pb-CpG to capture the smaller CNVs, we're able to achieve 100% recall. Next, I want to introduce our new phasing tool, HiFase. Historically, third-party tools such as WhatsApp have had good performance for small variant phasing and are well accepted by the community. And while this is encouraging, there are gaps in phasing capabilities that need to be addressed, including breaks in phase blocks caused by alignment issues, and phasing of other types of variants such as structural variants and tandem repeats. With HiFase, we're now able to address these gaps, increase performance and accuracy, and add a few other quality of life additions such as multi-threading and simultaneous BAM haplotagging and gathering of statistics. Specifically, HiFase takes in an alignment file from PBMM2 and variant calls from tools such as Deep Variant and pb-CpG to produce a phased VCF and haplotag BAM following the same convention as WhatsApp. And this allows users to upgrade to this new tool to enjoy all the improvement with minimal integration effort. HiFase demonstrates improved performance across the board, shown here on the left. We see HiFase demonstrates higher NG50, is able to fully phase more genes and variants, simultaneously phases structural variants, which cannot be done with WhatsApp, and leads to fewer switch flip errors. And finally, the resulting haplotag BAM file can be used by other downstream tools or pulled into IGV to easily separate reads by haplotype and visualize your phased variants. The last tool I want to mention is pb-CpG-tools for methylation statistics. So as I mentioned previously, HiFi sequencing is able to detect methylation without the need of a separate library prep. On Revio and Sequel IIe, the 5MC methylation calling is done on the instrument using our tool Jasmine, which adds MM and ML tags to the BAM file to indicate the position and probability of 5MC methylation across each read in your dataset. Following alignment of your data to the reference genome, pb-CpG-tools further performs a pileup to generate methylation probabilities for each site and haplotype. The output of pb-CpG-tools is a bed file to capture the methylation score for each site, and is accompanied with a bigwig file so users can pull in both the BAM and the bigwig file to visualize the methylation in IGV. Shown here is a pileup with reads separated by haplotype, demonstrating hypomethylation in haplotype 1 and hypermethylation in haplotype 2. This information is summarized by pb-CpG-tools in the bed files at the top of the pileup, demonstrating roughly 50 to 60% methylation in total for this site, but a dip in methylation in haplotype 1 and extremely high methylation in haplotype 2. With all of the advanced tools and all the different types of variants, the next reasonable thing to do would be to have a pipeline to streamline data processing and provide a comprehensive view of your HiFi sequencing data. Therefore, we've developed the PacBio WGS variant pipeline. The WGS variant pipeline connects all of the tools we just discussed, starting from alignment and including all the variant types, including small variants, structural variants, tandem repeats, segmental duplications, full phasing, copy number variants, and finally, methylation scores. It also optionally adds GLNexus for small cohort-based variant calling, and SLIVAR and SVPack to help users get started with variant annotation. The pipeline is written in workflow description language, or WDL, and is pre-configured on all major cloud platforms such as AWS or Azure to lead to lower effort for people to get started with their data analysis. However, this can also be run on-prem on local HPC systems. It expects 64 CPUs and 256 gigs of RAM. And with these resources, it can run a single 30x human genome in 17 hours or a trio in 22 hours. It's primarily distributed on GitHub as a command line tool, but you can also access the pipeline through our PacBio compatible partners whom offer more point-and-click solutions. If there's limitation for users to establish command line tools or looking for compute infrastructure for solid performance on data analysis, we have 4 partners which offer data analysis platforms on the cloud with ready-to-run WGS variant pipelines. These include DNAnexus, FormBio, Terra, and DNAstack. Additionally, last year, we also welcomed 2 tertiary analysis partners to provide services interpreting the whole genome sequencing variant calls, especially in rare and inherited disease areas. These include Genix and Golden Helix. So now with all of these advanced tools, pipelines, and effort from our partners, we have all the pieces together to be able to fully explore the human genome. But we aren't finished yet. We have a lot more planned in the next year. In addition to bug fixes and maintenance updates like improving platform compatibility, updating to newer versions of our existing tools, we'll also be adding new types of analyses. We plan on adding pharmacogenomic gene typing with our tools such as Pangu and pb-Starface. We have plans for calling homoplasmic and heteroplasmic variants in mitochondrial DNA. Team members are working on our next generation tool for structural variant discovery. And finally, we will add tools for the analysis of atypical or differential methylation. So keep an eye on our GitHub page for new releases. So with that, I'll just conclude with a few additional notes and resources. First and foremost, I want to let you know that PacBio can offer you an end-to-end solution from sample and library prep to sequencing and primary analysis on board our instruments. Once the data comes off our instrument, it is immediately ready to go for secondary analysis without any additional data processing needed. Therefore, today we focus some on our secondary analysis solutions such as SMRT-Link, GitHub tools, and our compatible partners such as DNAnexus, Stack, and FormBio. Finally, thanks to the addition of our new partners at Genix and Golden Helix, we also have a way to derive biological insights from your secondary analysis results. If you want to learn more, I recommend first checking out our WGS variant pipeline app note. If you want to try out any of these tools or experience PacBio HiFi data for yourself, we have a variety of demo datasets available on our website that you can freely download for all different types of applications. And if you want to try out or learn any more about our tools, you can find more information on our computational tools page or on each tool's respective GitHub repository. Finally, and not shown here, but worth mentioning, is visiting our page for our wonderful compatible partners. With that, I want to thank you all for watching, and please feel free to contact me or your local PacBio support team if you have any questions or want to discuss any projects. We are absolutely here to help at every step of the way, so get out there and start sequencing some HiFi genomes. Thanks for listening.